DNA synthesis, and we keep going there. And it's hard to do that and to be fresh because you've had it so many times. You've had it in biology in high school. But hopefully there'll be a few new things that you'll learn along the way here about DNA synthesis. We do need to cover it though. So don't forget, these are just showing oligonucleotides, RNA on the left and DNA on the right, the deoxy and the hydroxy group, and the difference between the two. A couple things you want to remember, that the backbone of DNA is the sugar phosphate, that's the sugar phosphate backbone that we talk about. And the bases are attached to the number one carbon of the ribose unit, right, to the individual bases. During DNA synthesis, this structure generally has a direction to it because the 5' of these guys have a phosphate on them. In the case of DNA, it's always a monophosphate. In the case of the RNA, it's usually, or often, is a triphosphate. Whereas on the other end, there's a 3' hydroxyl, and this 3' hydroxyl there is required for the enzyme. The enzyme has to have that 3' hydroxyl to add the incoming nucleotide triphosphate to. You can actually add one up on this end here. The problem is that DNA, the integrity of DNA synthesis is guarded very carefully by the cell because there's only 2 copies of that DNA. So when every time the cell replicates DNA, it has to proofread the nucleotide that it just put in to be sure it's the right one. And the only way it can do that is to add at this end here, right? Because if it adds the wrong one, the wrong nucleotide on this end, it can cut it off, right? And that cuts the nucleotide off, and that leaves a free 3' hydroxyl still on this end, so you can build on again. On the other hand, at this end here, if you cut off or like here, DNA, right? This guy here is a monophosphate. And remember, when nucleotides come in, you have the triphosphate that comes in and attaches to this hydroxyl, and that triphosphate is where the energy is involved for forming this bond down here. Does everybody understand that? On the other hand, if you come up here and you have the hydroxyl that you want to attach to that phosphate, it's a monophosphate. So there's no energy to attach it. So you always have to proofread at the bottom here, because if you cut one off, you always end up with that guy, with the hydroxyl group that you can add here. I'll try to remember to show you that again when we get there. So DNA replication, we said, is bidirectional. It goes in two directions. This is one half of the loop, the other half goes here. And in bacterial cells that have a round genome, of course, there's a single replication bubble. In eukaryotic cells, which have much longer chromosomes, the replication bubble is are more frequent. So on one single chromosome, there can be 1,000 bubbles, and there's several reasons for that. One is that the DNA polymerases in eukaryotic cells are much slower than those in prokaryotic cells. Prokaryotes, remember, go 1,000 nucleotides per second. Eukaryotes are lucky if they make 100. They're very slow. Why are they so much slower? Can we have an answer real quick? What's the difference between? Go ahead and yell.
Is there proofreading?
They are proofreading, that's true, but E. coli proofreads. That's where we discovered it. What's the difference between DNA in a prokaryotic cell and what we have in a eukaryotic cell? The DNA is packaged in chromatin, right? In bacterial cells, they're histone-like proteins, but they're really sort of free. So replication just goes. On the other hand, as we'll talk about in eukaryotic cells, that DNA is packaged very tightly in this nucleosome structure. And in order for polymerases to read through that, they have to deal with that structure first. So they're much slower. Anyway, there's a lot of these replication units. If you look at a replication, at each end of the bubble, this is called a replication fork. The polymerase complex is moving this way and opening up these parental strands, so it has to unwind it. And then DNA synthesis goes on. Now remember on the last slide, we said that DNA synthesis, polymerases have to have this 3' hydroxyl in order to add a nucleotide to. Well, if you look at DNA synthesis, right, as this strand opens there, this is the template strand, 3' to 5'. DNA is anti-parallel. It has to be anti-parallel in order for the bases to line up with the hydrogen bonds to form double-stranded DNA. Right, so this is the new strand, the pink, and it's anti-parallel. So if this is 3 to 5, this is 5 to 3. There's a 3' hydroxyl there. As this strand opens up, DNA polymerase can simply add on to that 3' hydroxyl. On the other hand, the other template strand is 5 to 3, which means that these new DNAs is 3 to 5, right? And parallel, but DNA synthesis always goes in the 5' to 3' direction, so that's why these arrows are in this direction. You see those going in that direction. In order to do that, what has to happen is that every time one of these pieces is made here, this strand has to open about 1,000 nucleotides or so. And when it does, the DNA is primed, a new fragment is made, right? And they're all made in this direction like that. Those are called Okazaki fragments. They're named after the husband and wife team, actually, who discovered these in E. coli. They isolated these pieces of DNA that were about 1,000 or 1,200 nucleotides long as the cell was replicating. If you have a question as we go, ask. I'm going to look, I'm just sort of reviewing through this since I think you've had most of that. So this strand has to open, and we have to have their DNA synthesis is initiated with a primer. All right, the things you need for DNA synthesis are nucleotide triphosphates, ATP, some sorts of energy, magnesium for the ATP, and a template and a primer. And why do you think you need a primer for DNA synthesis? When you make RNA, so when you make an RNA, you don't need a primer at all. So this guy here, you don't need a primer to make RNA, but you need a primer to make DNA. Does anybody have any idea why? I'll tell you again later, but one of the properties, the DNA polymerase is certainly the one that makes the bulk of the DNA, has 3 activities. It has the polymerizing activities, which adds nucleotides on, but then it has 5' exonuclease and 3' exonuclease. So if there's a single-stranded piece of DNA that has a loose 3' end, it can cut it off. And that 3' activity is a proofreading activity, right? That's how it checks to be sure if this nucleotide is the one that it just added is the correct nucleotide. How does it know if it's the correct nucleotide? How does it know if it's correct? Well, remember what DNA synthesis is, right? This guy's there, you have a template that's acting as a template for this. So the last nucleotide that's put in should be complementary to the base over in the template, right? And if they're complementary, what do? They hydrogen bond. They form hydrogen bonds between the bases. If they're not complementary, what don't they do? They don't form hydrogen bonds. So DNA polymerase actually looks at the last nucleotide it puts in and sees if there's a hydrogen bond. If not, the 3' to 5', this is the 3' ends in this direction, 3' to 5' exonuclease cuts off that free hanging base or nucleotide and tries again, looks for the right one. Make sense? Okay, so because of that, proofreading activity, right, DNA requires a primer. It's an absolute essential property of DNA polymerase, that proofread. If you were to list the steps of DNA polymerization, the first one would be proofreading. So the polymerase proofreads to be sure the nucleotide's right, then it reads the next template nucleotide, then it selects the proper one, and then it makes the bond, makes that the sugar phosphate bond, that phosphodiester bond, right? Does everybody see that? You have a question? Do I have to draw it? Okay, so proofreading requires, because of that, DNA synthesis always has to have a primer. You have to have a primer here so that the polymerase can proofread the last nucleotide in that primer before it adds a new one. How long is the primer? How long is the primer? About 10 nucleotides. Why is it 10 nucleotides? Who works in the lab? When you make a hybrid, or you make complementary DNA, 2 strands of oligonucleotides, and you let them complementary base pair. Right? There's no stability unless there's about 7 nucleotides, 7 or 8. Then the 2 will hybridize. If there's less than that, they just pop apart. There's no stability in the hybrid. So to have 10 is just about right? It's a very when that primer interacts with the DNA, it binds sort of tightly, tight enough to stay there. Okay? What's the primer made out of? RNA. Why is it made of RNA?
Because it can bind to single-stranded DNA.
Well, so can DNA. But why is it made of RNA? Because DNA polymerase can't take a template and put down a piece of DNA, right? It can't do that. It has to proofread. So the only thing that can take a template and make a primer is an RNA polymerase, right? A DNA-dependent RNA polymerase. DNA-dependent because it has a DNA template. RNA polymerase because it makes a piece of RNA about 10 nucleotides long and serves as a primer, right? And then you can synthesize. Now that primer's there, DNA polymerase can proofread it, no problem, right? Okay. So now we've made these Okazaki fragments. Let me see if I have that somewhere. But these Okazaki fragments are made like this, right? And they have to be corrected. This is essentially a DNA error. So the DNA has to correct it. And actually the polymerase that corrects it is the same polymerase that corrects damage to DNA, damaged DNA. Okay, the same polymerase. How do you have to correct this? There's two things that have to be done to correct it, right? What's the first thing? What's the first thing? So all of these guys, right? Each one of these has a little primer, 10 nucleotides of RNA and then 1,000 of DNA. 10 nucleotides of RNA, 1,000 of DNA. So what do you have to do? To make these things, this DNA strand intact, you have to do two things. You have to get rid of the RNA, and you have to put the DNA fragments, all those Okazaki fragments, back together again, right? You have to glue them together. You remove these guys, right? What direction are these guys? This is the 5' end and the 3' end. So this is the 5' end of that new DNA, right? That's where the RNA primer is. You want to cut it off in this direction, so what activity do you need? You need a 5' to 3' exonuclease. What's the difference between an exo and an endonuclease? Exo cuts on the ends, right, and an endo cuts in the middle. The proofreading is a 3' to 5' endo. This new chain is going this way. This is the 3' hydroxyl. So the DNA polymerase is proofreading that way, 3' to 5. But to get rid of the RNA, it needs a 5' to 3' exonuclease that cuts that out. When it cuts it out, what happens? It leaves a hole of about 10 nucleotides, and what's on the opposite side of that hole?
RNA.
A template. No, a DNA template, right? You cut this piece of RNA out, this is the DNA template there. So now that serves as a template, so once it's cut out, then DNA polymerase comes in and fills that in with a piece of DNA. Right? Fills the gap in with DNA. And once you fill in the gap, so now these are all solid DNA, then what do you have to do?
Glue them together, stick them.
Put a ligase. You have to have DNA ligase to put these together because DNA polymerase cannot take a 3' hydroxyl and a 5' phosphate and put them together. Why can't it? Why can't it do that? 3' hydroxyl super, what's wrong with that 5' phosphate? Monophosphate, no energy for the reaction. Right? It has to be a triphosphate. And actually what polymerase, I don't remember if I'm going to tell you about ligase, but what ligase does is it takes that 5' phosphate and it adds another phosphate to it. So now there's a phosphodiester bond, there's energy there, and as that bond cleaves, they can form a bond with the hydroxyl group. So that's all ligase does, right? Interestingly, if I showed you this, let me turn this on real quick. If you look at how this happens, I'm just going to pause.